All right, so let's move back to Sungsik's talk. Sunsik, can you try
Yes,
to share the screen again?
I will try one more time.
Yeah, that's it.
All right, can everybody see the
Okay,
screen now?
perfect.
Awesome, great, yes.
Okay, great. Okay,
Thank you very
so
much.
we have Sungsik Kon from Ohio State University. He will be talking about comparative performance of popular methods for hybrid detection using genomic data. Sungsik, please.
All right, so good morning or good evening, everyone. First, I would like to apologize for the technical difficulty I had. My name is Sungsik Kevin Kong. I'm a PhD student at the Ohio State University. And thank you very much for this opportunity for me to participate. And I'm really glad to share one of my dissertation chapters with you today, entitled Comparative Performance of Popular Methods for Hybrid Detection Using Genomic Data, which has been recently published in Systematic Biology. So hybridization is an important biological process that can result in the production of novel evolutionary lineage and lateral movement of genetic materials between species. However, this process has been much neglected in comparison to traditional bifurcating speciation process, because the product of hybridization is not as easy to observe as in vertical speciation, although now we realize it is much more common in nature than we previously thought. Even for the organisms where hybridization is suspected, identifying them based on phenotype is impractical, because expectation that the hybrids will possess intermediate phenotypes of their parents is not always true. But fortunately, accumulation of genomic data and advances in statistical and computational methods now allow us to accurately detect hybridization. So one way to detect hybridization is to construct a phylogenetic network. Phylogenetic networks are distinguished from the phylogenetic trees, which basically describes an evolutionary history of organisms by a reticulation structure, where two independent lineages merge into a single node that illustrate non-tree-like scenarios. And each reticulation edge has an inheritance parameter called gamma that can be biologically interpreted as the proportion of genomic contribution from one of the parents to the hybrid daughter. And there are a handful of tools that can infer phylogenetic networks, but unfortunately, they all lack scalability, meaning that they only can cope with the small size data. So alternatively, people use non-network methods to detect hybrids when they have genomic data set in hand. In general, these methods can be largely categorized as site-parent frequency-based methods that includes Patterson's D-statistic, which is also called the ABA-BABA test, and HIDI, and population clustering methods are represented by the Bayesian-based structure and the maximum likelihood-based admixture. So site-parent frequency-based method conducts a formal hypothesis test for the presence of hybridization. And in brief, suppose we have 4 taxa, which we know that their most likely relationship is the tree as shown on the far left. And for the 3 trees on the right, we basically substituted each taxon name for each site on their sequence. So A and B can be any of the 4 nucleotides, A, C, T, or G, but they're not equal to each other. And the method will basically count how many times we observe such patterns shown here in the 2nd, 3rd, or 4th tree. So in the lack of hybridization, we would expect to see the 2nd pattern most frequently, and the probabilities of observing the patterns ABA or BABA, which are discordant to the true relationship, should be basically equal if they solely arose due to incomplete lineage sorting instead of hybridization. However, when 2 patterns, ABA and BABA, occur in significantly different proportions, we interpret such pattern is arose due to hybridization. So both HIDI and the D statistic follow very similar philosophy, but HIDI also utilizes the counts of BBAA, which is the 2nd pattern, which is again to estimate gamma, which is again the proportion of genomic contribution from parent to the hybrid daughter. And in contrast, the clustering methods are popular for detecting hybrids on population level, probably because it can cope with many individuals simultaneously instead of 4 we see in the previous slide. So briefly, usually we specify the number of expected genetic groups in the dataset, which we call kappa. And the method estimates what is called ancestry coefficient, which tells us the probability of genetic material inherited from the assumed gene pool to the contemporary individual. And the final result is usually summarized and visualized as shown in the figure here, where each vertical bar represents each individual, and different colors basically represent the probability of the individual belong to the different gene pool. So for example, here, the researchers assumed kappa is equal to, so we see two colors, meaning that two genetic pools contributed to all of the individuals in the sample. And they identified pure and hybrids based on the value of ancestry coefficient. The issue here with this method is that there's no objective way to interpret the coefficient. And most studies basically arbitrarily set their threshold to decide if the population is pure or hybrid. And in the most case, it is difficult to decide, and often researchers label many individuals as ambiguous, as we see in group 4 here. And moreover, it is important to stress that the ancestry coefficients are not the same as gamma, although they are being used indiscriminately in practice. So in this study, our goal is to comparatively evaluate the performance of the discussed 4 common methods for hybrid detection using simulation in a wide range of hybridization scenarios. And we discuss their appropriateness in hybridization studies. So let me quickly go over the methodology. So we consider 6 hybridization scenarios. From the top left, a single hybrid speciation, and then a single integration scenario in B, which is distinguished from the single hybrid species scenario by having the hybrid lineage completely merged to one of its parents via backcrossing. And we have double and triple independent hybridization scenarios, C and D, and 3 hybridizations then overlap, meaning the ancestor involves more than a single hybridization event to give more complexity. And finally, complex mixture of recent and ancient hybridizations. However, in this presentation, I'm going to focus mainly on the top 3 scenarios in detail due to time limitation. So we simulated all of our data set to accurately evaluate the method's performance in different biological conditions. In a single hybrid speciation scenario, we manipulated gamma ranging from 0 to 0.5 in an increment of 0.1, where 0 would represent lack of hybridization and 0.5 represents equal genomic contribution from both parents. And we have 3 branch lengths where tau 1 is more recent than tau 2 or 3, as we can see on the network. And we set tau 1 from 0.05 to 5, where tau 1 represents time elapsed since hybridization, while keeping tau 2 as 1.5. And we set tau 2 ranging from 0.05 to 5, while setting tau 1 fixed to 1.5 to evaluate the method's performance in different amount of incomplete integer sorting, which I will call ILS from now on in short. So what we expect here is that the data created with short tau 2s will have high ILS, since the gene trees would fail to coalesce in such a short speciation time. And this is important because hybridization and ILS leaves very similar footprint in an organism's genome. And it is crucial for the methods to differentiate them in order to correctly detect hybridization in presence of ILS. And we set tau 3 to 5, which is long enough to confidently segregate the in-group taxa P1H and P2 from the out-group. In total, we have 54 combinations of parameters for the single hybridization scenario. For the rest of the scenarios, we used a single set of parameters by setting gamma to 0.5 and all taus to 1.5, except the one that is linked to the root. Each population had multiple individuals, 10 for each parent, 5 for hybrid, and 1 for the outgroup. And we randomly picked a single individual from each population for the D statistic, since it can only work with 4 individuals. And for each scenario, we broke down the network into 2 species trees or more by removing one of the reticulation edges and simulated 1,000 gene trees for each species tree in the proportion of gamma. So what I mean here is, for example, if our gamma was 0.5, we would broke down this phylogenetic network on the left into 2 species trees we see on the right, and 500 gene trees were simulated for species tree 1, and another 500 for 2, and together we simulate 1,000 gene trees. And for each gene tree, we simulate a sequence that is 100 base pairs long to mimic short read length in next generation sequencing method. And we finally concatenated all the sequences for each taxon to create a full alignment. And for each alignment, we conducted Heidi analysis and we recycled the site pattern counts from Heidi to compute the D statistic. And then we ran structure and admixture analysis using the same data set following the general protocol employed in practice. But we manually selected kappa by looking at the true number of parental populations in the scenario in interest. And we made 100 replications through Unity cluster at the Ohio State University. And finally, we looked into a number of factors to evaluate the performance of each method. First, we evaluate the power, which tells us how often the method correctly rejects the null hypothesis, which can be interpreted as correct detection of hybridization. And strictly speaking, since the power requires a hypothesis testing, the rate of identifying hybrids in structure and admixture analysis cannot be referred to as power. But we include them here anyway for the comparison purposes. And second, we evaluate the accuracy of estimates of gamma by computing the mean squared error, which is basically the distance between the estimates and the true value. And finally, we conducted clump-peg analysis to summarize and visualize population clustering analysis. So these bar graphs present the evaluated power of different methods labeled in various colors on top in different conditions in the hybridization scenario. On the left side, we modified tau1, and as we moved downward, tau1 was increased in the first number in the parenthesis while keeping tau2 to 1.5. So as we move down on the left side of the graphs, this means time elapsed since hybridization is increasing. And on the right side, we increase tau2 while keeping tau1 constant. So the amount of incomplete sorting is the greatest on the top, and it decreases as we move downwards. Within each plot, gamma increased from left to right, and the bars at far left represent type 1 error, since they detected hybridization when the gamma value was 0. So the first thing to notice here is that we don't see as many red bars as the other colors, meaning that the detection power in admixture was not as good as the rest, particularly when gamma was small and when the amount of ILS was high. In contrast, the power of Heidi and the D statistic, colored in gray, blue, and yellow, was very high in the most cases, regardless of the value of gamma, except when the amount of ILS was excessively high. And lastly, structure, colored in green, performed relatively better than admixture, although it still showed poorer performance when the gamma was small. And something to note for structure here is that even though it identified hybrids the most frequently when the amount of ILS was excessively high, this must be carefully interpreted because it may not represent that structure can detect hybridization despite high amount of ILS, but it simply cannot distinguish the two processes since it had also very high type 1 error. These plots present the accuracy of gamma estimations in admixture on top, Heidi, and structure. And as we move from left to right in the first 5 columns, the tau1 was increased, and the numbers on the parenthesis. Tau1 was increased while keeping tau2 constant, followed by 4 plots on the right that keeps tau1 constant while tau2 was increased. Accuracy of the estimates gets better as the points get closer to the black diagonal line, or if the mean squared error, which is represented in the black solid line with dots on the bottom, is close to the x-axis. First, we noticed that the plots for admixture is very spacious, and this is because they simply failed to estimate any. And however, estimates of gamma was not horrible when they actually worked. Moving down to the bottom row, we can see that structure identified hybrids much more frequently than in admixture, but in many cases, their estimates were far from the true value. And what we also notice here is that the estimates tend to form multiple clusters, which reflects the phenomenon known as the multimodality, which can lead to serious misinterpretation in biological sense. And lastly, we observed that estimates in Heidi are very accurate in general in the middle row, except in this plot in the middle where the amount of ILS was excessively high. Not only the power was very low in this case, but estimates of gamma were very inaccurate. However, this issue could be fixed when we increase the data set by 10-fold, which is on the left side, when Heidi began to detect hybrids much more frequently despite high ILS, although their accuracy was not yet ideal with high type 1 error. When we increase the data set size to 100-fold, which we see on the right side, Heidi began to detect hybrids with accurate estimates of gamma, meaning that its failure to detect in the previous slide is not due to its algorithmic limitation, but simply it needed more information. And for the introgression scenario, admixture completely failed to identify any hybrids, but Heidi and structure performed very well. And the plots on the left show their estimates of gamma, and they were fairly accurate as reflected in low value of mean squared error, which is a small black dot on the bottom. And structure, but the estimates in Heidi was not as good as the single hybrid speciation scenario we see previously. And structure estimates continue to show this multimodality issue. So we looked into the reason for decreased performance in Heidi and produced data set by varying the time of introgression. And we report on the right side that the accuracy of gamma in Heidi could be improved in introgression scenario when the gamma was asymmetric, which means it is small, and when the introgression happens further away from the split event. And in the scenario with two independent hybridization, estimates of gamma for both reticulation edges in Heidi were outstanding, as we can see on the top left plot. And for the population clustering methods, we ran Klumpek analysis to summarize and visualize the outputs, and color coded here as red for admixture and green for structure analysis. And each block within the plot, which within the Klumpek plot, represents a distinct population. And the red line indicates the true parental population for the hybrid populations. So we would expect every parent in different color and exactly half of those colors in the hybrid populations. And we assumed kappa to be 4 since there should be 4 parents in total. And admixture replicates were summarized into 2 distinct solutions in E and F, where one solution correctly identified both hybrid populations and their parents, when the other only identified only one hybrid population. For the structure analysis, we first assumed kappa to be 4 and identified 3 optimal solutions shown here, B, C, and D in lighter green plots. However, most hybrid populations were not clearly identified, and even if when they were, they contained more than 2 ancestries. And we thought that this might be because the analysis assigned the outgroup as a distinct population. So we repeated the analysis with increased kappa to 5, and which the results are shown in darker green plots on G, H, I, and J. So where the hybrid populations were not identified in 3 out of 4 solutions, and even for the one that actually identified population admixture, they had 3 ancestries, making biological interpretations difficult. So in this study, we have shown that Heidi is very reliable when it comes to hybrid detection. However, we also report that high amount of ILS can reduce both detection power and accuracy of gamma estimates. Also, although this may not be as severe as before, ancient hybridization can also reduce the accuracy of gamma estimates, although both issues can fade away with increased dataset size. The bigger problem here is that since the model implemented in Heidi assumes hybrid species scenario, the estimates may not be accurate if the populations experience integration in nature, which might occur more frequently in empirical systems. And finally, should we continue to use the population clustering method for hybrid detection? We still believe the clustering methods are in fact extremely useful for population level studies because phylogenetic network analysis has scalability issue and the site pattern-based methods requires large data set size, which is not always handy. However, interpretations from the clustering analysis must be carefully conducted, as we observe that identification of hybrids based on ancestry coefficient can be subjective, and intermediate ancestry coefficient does not necessarily indicate historical hybridization. And finally, absence of admixed population is not evidence of absence of hybridization. So this ends my presentation. Thank you very much for listening, and I would appreciate any question or feedbacks.
Okay, thank you very much. So okay, now we can take a couple of questions. Okay, I have a question. So this study is more like from practical, like a theoretical simulated data. I wonder how often in reality those hybridization occur. Let's say in one branch of bacteria, like around E. coli, are there lots of hybridization going on in the past?
Yes. So this really depends on the system. So when we talk about bacteria or more small cell organisms we call that usually horizontal gene transfer instead of hybridization. So in this study, we focus on the hybridization, which is basically a merge between two different species. And this usually happens in plants and animals. So the focus was plants and animals. And the frequency of hybridization in nature, previously we thought this is really rare phenomenon, but then the studies comes out and it's really, the hybridization studies are very popular recently. And we realized that more than 20% of plants have arose due to historical hybridization, and around similar amount among the birds. And even in the animals, we thought it's really rare, but it's actually much common than we previously thought.
Oh, okay, very interesting. Okay, Sungsik, thank you very much again. And I think you mentioned that you did a thesis, so you finished. You are now a Dr. Con, I guess.
Not yet. So
oh, not
I'm
yet. Okay,
not
close,
yet.
Close. All right, close. Okay.
I'm still working on writing my dissertation,
Okay.
but
Good luck. Good luck for that.
Thank you very much.